Hey everyone, welcome back. Ready for another deep dive?
Definitely. Always ready to geek out about some cool science.
Well, today we're dissecting episode 15 of Cocktail Party Chemistry, cutting-edge biosensors and simulations.
Sounds intense.
Oh, it is, but in a good way. Think mind-blowing science, perfect for sparking conversations.
Like, what exactly are we talking about here?
We're diving into 5 fascinating areas.
Hit me with them.
First up, biosensors. They're basically nature's blueprint for detecting the tiniest things.
So, like, super-powered senses.
You could say that. Then there's molecular dynamics. Think virtual labs giving us atomic-level insights.
Whoa, getting futuristic already.
And we're just getting started. We've also got DNA origami, folding DNA into incredibly tiny devices.
DNA origami. Like, what? Folding those double helix ladders into tiny creams?
It's not that far off.
Yeah.
Then we're going to look at electrochemical sensors. Rapidly detecting pollutants, super important these days, right?
Yeah, definitely can't hurt with all the environmental concerns we're facing.
Exactly. And lastly, we're going to explore self-assembling nanostructures. It's all about witnessing order emerging from chaos at the nanoscale.
Order from chaos sounds a bit philosophical for a science deep dive.
Well, you'll be surprised how much philosophy there is in science sometimes. But before we get too philosophical, let's start with those biosensors. What's got you intrigued about those from the episode description?
Honestly, the idea of taking inspiration from nature's own detection systems, it's like we're finally catching up to what nature's been doing for millions of years.
Right. It's pretty amazing. But I'm curious, what are the different types of biosensors, and how are they actually being used out in the real world?
Oh, tons of different types, each designed to detect a specific thing. But let's start with one of the most common enzymatic biosensors.
Okay, enzymatic biosensors, lay it on me.
These guys rely on the super specific nature of enzymes. Imagine an enzyme as a lock and its target molecule as a key.
So only that one key can unlock the lock.
You got it. And that unlocking triggers a reaction we can detect.
Okay, so it's like a super picky molecular guard dog sniffing out a specific target.
Exactly. Think about those blood glucose monitors for people with diabetes.
Oh yeah, my grandma uses one of those.
Well, that's an enzymatic biosensor. It uses an enzyme that reacts with glucose specifically, and the signal it creates tells you how much glucose is present.
Whoa, personalized medicine on a molecular level.
It really is. But it's not just blood sugar. Enzymatic biosensors are used to detect all sorts of things: environmental pollutants, food contaminants, even disease markers in medical tests.
So these little molecular detectives are out there protecting our health and the environment.
You got it. They're everywhere, working behind the scenes. But let's move on to another type, immunosensors. These guys use antibodies. You know, those proteins our immune systems use to fight off invaders.
Right, the little warriors that latch onto specific targets and neutralize them. But wait, how do we turn an antibody into a sensor?
Okay, picture this. You have an antibody that specifically binds to, let's say, a certain bacteria. You attach that antibody to a sensor surface.
Okay, I'm following. Then what?
Now, when that bacteria comes along, bam, it binds to the antibody.
And that triggers.
A detectable change. It could be a shift in light, a change in electrical current, even a change in mass. There are all sorts of clever ways to translate those tiny molecular interactions into signals we can measure.
So we're like eavesdropping on these molecular conversations, picking up on their subtle signals to understand what's happening. Pretty cool.
It is. And this opens up a whole world of possibilities for things like rapid disease diagnostics. Imagine a device that can tell you if you have the flu in minutes, just from a saliva sample.
Oh, man. No more agonizing over whether it's a cold or something worse. Talk about convenience.
Oh.
But what else are these immunosensors being used for?
Oh, so many things. Cancer cell detection, toxin detection in food, even for sniffing out biological weapons.
Whoa, seriously. Okay, so we've got enzymes and antibodies working as molecular detectives. Any other types of biosensors out there?
You bet. Remember those DNA origami structures we talked about? Well, we're also building sensors out of DNA.
Hold on. DNA sensors. This is getting seriously futuristic.
It is. These DNA biosensors, they rely on that super specific binding of complementary DNA strands. Let's say you have a single strand of DNA designed to bind to a specific sequence in a bacteria.
Okay, so kind of like a DNA lock that only a specific DNA key can open.
Yeah, exactly. You attach that DNA strand to a sensor surface, and if that bacteria is present, its DNA will bind to the sensor DNA, triggering a signal.
So it's like a microscopic DNA handshake that sets off an alarm. What can we detect with these DNA biosensors?
Oh, tons of things. Genetic mutations, pathogens in water, even genetically modified organisms in food.
Seriously, this is mind-blowing. We're talking about a level of precision that was unthinkable a few years ago.
It's a rapidly evolving field, constantly pushing the boundaries of what's possible, and it's going to have a huge impact on our lives.
Okay, so much incredible stuff already. But before we get too carried away, are there any downsides? Like ethically, these technologies seem super powerful.
That's a really important point. Power like this, it comes with responsibility.
What kind of ethical concerns are we talking about?
For starters, data privacy. These biosensors generate tons of personal health information. We have to make sure that information is protected and used ethically.
Right. Wouldn't want our personal health data falling into the wrong hands? What else?
Well, there's the potential for misuse too. What if employers start using biosensors to screen job applicants for genetic predispositions to certain diseases?
Oh, wow. Yeah, that's a bit scary. Or insurance companies using that info to deny coverage.
Exactly. We need to have serious conversations about all of this as these technologies become more widespread.
Absolutely. We need to establish clear guidelines and regulations. But before we get bogged down in the what-ifs, let's stay focused on the science for now. What do you say we dive deeper into the world of biosensors, starting with those enzymatic ones?
Sounds good to me. Let's explore how scientists are harnessing nature's genius to detect the tiniest things.
All right, let's do it. I'm ready to learn more about how these microscopic detectives are changing the world.
So, we were diving deep into the world of biosensors. Remember those enzymatic biosensors? They use enzymes, those picky little proteins, to detect specific molecules.
Right, like a lock and key system. Only the right molecule can trigger the enzyme.
Exactly. It's incredibly precise. But enzymes aren't the only players in the biosensor game. We also have immunosensors.
Oh yeah, those use antibodies, right? Those protein warriors that our immune systems use to fight off invaders.
You got it. And they work in a similar way. Let's say you want to detect a specific bacteria.
Okay, how do we do that with an antibody?
You take an antibody that binds specifically to that bacteria and attach it to a sensor surface.
Gotcha, so when that bacteria comes along.
Bam! It binds to the antibody, setting off a detectable change. Could be a shift in light, a change in electrical current, even a change in mass.
So we're basically translating these tiny molecular interactions into signals we can actually measure.
Exactly. It's like we're eavesdropping on the molecular world, using antibodies as our listening devices.
That's pretty cool. So what are some real-world applications for these immunosensors?
Well, one of the most exciting areas is in rapid disease diagnostics. Imagine getting a flu diagnosis in minutes just from a saliva sample.
No more waiting days for those lab results. That would be a game changer.
Absolutely. And that's just the tip of the iceberg. Scientists are developing immunosensors for all sorts of things, from detecting cancer cells to sniffing out toxins in food.
Whoa, they really are like little molecular detectives.
They are. But wait, there's more. Remember those DNA origami structures we talked about? Well, guess what?
Don't tell me we're building sensors out of DNA now.
You got it. DNA biosensors.
This is getting seriously sci-fi. How do those even work?
Well, DNA has this incredible ability to bind to complementary strands with amazing specificity. So you design a single strand of DNA that's tailor-made to bind to a specific sequence in, let's say, a virus.
So it's like a DNA lock that only a specific DNA key can open.
Exactly. You attach that DNA strand to a sensor surface, and if that virus is present, its DNA will bind to the sensor DNA, triggering a signal.
So it's like a microscopic DNA handshake that sets off an alarm.
You got it. And the applications for these DNA biosensors are incredible, detecting genetic mutations, identifying pathogens in water, even spotting genetically modified organisms in food.
Wow, the level of precision is mind-blowing. It's like we're entering a whole new era of molecular detection.
We are, and it's just the beginning. The field of biosensors is constantly evolving, with new types of sensors being developed all the time. It's going to have a huge impact on our lives.
All this is amazing, but like we talked about before, we got to think about the ethical side too, right? This tech is incredibly powerful.
Absolutely. With great power comes great responsibility, as they say.
So data privacy is a big one, right? We've got to make sure all that personal health information from these biosensors is protected.
Definitely. And we also need to think about potential misuse. What if employers or insurance companies start using biosensors to discriminate against people?
Yeah, that's a scary thought. We need to be having those conversations now before these technologies become even more widespread.
Absolutely. We need to establish clear guidelines and regulations. But for now, let's shift gears and explore another fascinating area from the Cocktail Party Chemistry episode.
Okay, what's next on our scientific adventure?
Let's dive into the world of molecular dynamics. Remember those virtual labs we talked about?
Yeah, the ones that give us atomic level insights? I'm ready to enter the matrix of molecules.
All right, buckle up. Imagine you're a scientist trying to understand how a new drug interacts with a protein in the body. It would be incredibly challenging to observe this in a real lab. Molecules are tiny and their interactions are super fast.
Yeah, like trying to watch a hummingbird flap its wings in slow motion.
Exactly. That's where molecular dynamics comes in. We create computer models that simulate the behavior of those molecules.
So we're basically building a virtual world where we can zoom in and see everything in detail.
Precisely. We can see how individual atoms are moving, how bonds form and break, how molecules interact. It's like having a front row seat to the molecular dance of life.
Okay, I'm getting less Matrix and more Fantasia now.
Yeah.
But I'm definitely intrigued. What are some real-world applications of this technology?
Well, one of the most promising areas is drug discovery. By simulating how drug molecules interact with their targets, we can identify the most promising candidates and optimize their design.
So we're talking about using computer simulations to design better, safer drugs.
Exactly. And it's not just limited to pharmaceuticals. Molecular dynamics is being used to design all sorts of things, from new materials to sustainable energy solutions.
Okay, my mind is officially blown. It sounds like we're going from observing the molecular world to actually engineering it.
We are, and it's also helping us to understand the fundamental processes of life, like how proteins fold into their intricate 3D shapes.
Right, because misfolded proteins are linked to all sorts of diseases like Alzheimer's and Parkinson's.
Exactly. So by understanding how proteins fold, we can potentially develop new therapies for these devastating diseases.
So it's not just about designing new things. It's about understanding the very essence of life itself.
It is. And it's important to remember that these simulations are just that, simulations. They're powerful tools, but we need to be careful about the data we feed them and the assumptions we make.
Right, like any tool, they can be used for good or for bad. We need to be thoughtful about how we apply this technology.
Absolutely. But the potential benefits are immense, and I believe that this technology will ultimately help us to create a better world.
Okay, I'm sold. Molecular dynamics is definitely something to keep an eye on. But let's shift gears again and revisit another fascinating topic from the episode DNA Origami. Those tiny self-assembling structures are still on my mind.
Absolutely. Get ready to dive back into the world of molecular origami.
Lead the way. I'm ready to fold some DNA.
All right, let's recap the basics. DNA is that long, double-stranded molecule that carries our genetic code, and each strand is made up of those 4 building blocks: adenine, thymine, guanine, and cytosine.
Yeah, the famous A, T, G, and C. I remember learning about that in high school biology. But how do we go from genetic code to building materials?
The key is that A always pairs with T, and G always pairs with C. It's like a molecular puzzle, where each piece fits perfectly with its partner. And scientists can exploit that predictable pairing to design DNA sequences that will bind to each other in very specific ways, creating all sorts of shapes and structures.
So it's like having a set of molecular Lego blocks that we can use to build whatever we want.
You got it. And that's what DNA origami is, using DNA as a building material to create things at the nanoscale.
But how do we actually do that? How do we fold DNA into these intricate shapes? It sounds incredibly difficult.
It is complex, but the principle is surprisingly simple. Scientists start with a long strand of DNA called the scaffold strand. Then they design shorter strands called staple strands that are complementary to specific regions of the scaffold. When you mix these staple strands with the scaffold, they bind to their complementary regions, causing the scaffold to fold into a predetermined shape.
So it's like we're using the staple strands to stitch the scaffold strand into a specific pattern.
Exactly. And by carefully designing the sequences of the staple strands, scientists can create all sorts of shapes and structures: squares, triangles, tubes, even 3D objects.
Okay, my mind is blown again. But what's the practical application here? Is this just a cool science trick, or are we actually building things with DNA origami that could be used in the real world?
This is where it gets really exciting. DNA origami has enormous potential in a wide range of fields. One of the most promising areas is in medicine, specifically drug delivery.
Oh yeah, I remember hearing about that, like building tiny DNA cages that can carry drugs directly to specific cells.
You got it. That's one of the big goals, to create targeted drug delivery systems that can revolutionize the treatment of diseases like cancer.
That's incredible. It's like having a microscopic postal service that can deliver medicine right to the doorstep of diseased cells.
Exactly. But drug delivery is just one application. Scientists are also using DNA origami to create nanoscale sensors, build tiny circuits from molecular computing, and even create templates for the growth of new materials.
Whoa. It's like we're witnessing the birth of a new era of nanotechnology where DNA is the building block of the future.
We are, and it's an exciting time to be involved in this field. The discoveries are coming fast and furious.
Okay, I'm officially on board the DNA origami train.
Yeah.
But we've still got two more topics to cover from the cocktail party chemistry episode. Let's move on to those electrochemical sensors, those rapid pollutant detectors that you mentioned earlier.
Excellent choice. Get ready to dive into the world of electrochemistry, where chemical reactions are harnessed to create powerful sensing technologies.
I'm ready. I'm eager to learn more about how we're using chemistry to protect our environment and our health.
Okay, so electrochemical sensors, they're all about converting chemical information into electrical signals we can measure.
Right, kind of like translating the language of chemistry into the language of electronics.
Exactly. And it all comes down to these things called redox reactions. They involve the transfer of electrons between chemical species.
Okay, I vaguely remember redox reactions from chemistry class. But remind me, how does this electron transfer actually work in a sensor?
All right, let's say we want to measure the amount of a certain pollutant in a water sample.
Okay, I'm picturing a dirty lake.
Perfect. So we use an electrochemical sensor that contains a material that reacts with that specific pollutant.
So it's designed to target that one pollutant.
Exactly. When that pollutant hits the sensor, it triggers a redox reaction. Electrons start flowing,
And that flow of electrons.
creates an electrical signal that we can measure. The stronger the signal, the more of that pollutant is present.
Pretty slick. So where are these electrochemical sensors being used?
Oh, everywhere. One of the biggest areas is environmental monitoring. They're used to detect all sorts of pollutants in air, water, and soil.
So they're like little guardians of the environment.
You could say that. They're helping us track pollution levels, identify sources, and ultimately protect our planet and our health.
That's awesome. Give me some specific examples. How are these sensors actually being used out in the field?
Well, think about air quality monitoring. These sensors can detect harmful gases like carbon monoxide, nitrogen dioxide, and ozone.
Right, those are some nasty pollutants.
They are. And the data from these sensors can be used to issue public health warnings, help us understand pollution patterns, and even guide policy decisions.
Wow, so they're not just detecting pollution, they're helping us do something about it.
Exactly. And it's not just air. These sensors are also used extensively in water quality monitoring. They can detect heavy metals, pesticides, all sorts of nasty stuff that can contaminate our water.
It's amazing how these sensors are helping us protect our most precious resources. They're like the silent guardians of our environment.
They are. And their role is only going to become more important as we face the challenges of climate change and pollution.
Okay, so we've covered biosensors, molecular dynamics, DNA origami, and electrochemical sensors. We're on a roll. Just one more fascinating topic to go from our cocktail party chemistry episode. Self-assembling nanostructures.
Yes, order from chaos. It's a pretty mind-blowing concept.
It is. Remind me again how this whole self-assembly thing works.
So self-assembly is basically a process where molecules spontaneously organize themselves into ordered structures. It's driven by these weak interactions between the molecules.
So it's like they're naturally attracted to each other and just fall into place.
Exactly. Think of it like a perfectly choreographed dance, but at the molecular level.
I love that analogy. So what kind of structures are we building with self-assembly?
Oh, all sorts. One classic example is the formation of micelles. Those are tiny spheres that form when certain molecules are placed in water.
What's so special about my cells?
Well, they have all sorts of cool properties and can be used for things like drug delivery and even in cleaning products.
Okay, so we're talking tiny but powerful. What else?
Another cool example is liposomes. They're similar to micelles but have a bilayer structure like a cell membrane. They're being explored for drug delivery and gene therapy.
Wow, it's like we're building tiny artificial cells.
It really is, but the applications of self-assembly go far beyond that. Scientists are using it to create new materials with unique properties, like strength, flexibility, or conductivity. They're even using it to build templates for tissue engineering.
So we're talking about building materials that practically assemble themselves. That's incredible. What are some of the challenges with this technology?
One of the biggest challenges is control. It can be tricky to get the molecules to arrange themselves exactly the way we want.
Right, it's like trying to herd cats at the nanoscale.
Exactly. But scientists are getting better at it all the time, developing new techniques for manipulating self-assembly with incredible precision.
Wow, the possibilities seem endless. Well, I think we've covered a lot of ground today. Biosensors, molecular dynamics, DNA origami, electrochemical sensors, self-assembling nanostructures. It's been quite a journey.
It has, and it really highlights how science and technology are constantly pushing the boundaries of what's possible.
Absolutely. But before we wrap things up, it's important to remember that with all this incredible potential comes responsibility. We need to think about the ethical implications of these technologies and ensure they're used for good.
Couldn't agree more. We need to be mindful of things like data privacy, potential misuse, and equitable access. We need to make sure these advancements benefit all of humanity, not just a select few.
Absolutely. It's a conversation we need to be having now as these technologies become more integrated into our lives.
Exactly. But let's not lose sight of the incredible potential these technologies hold to solve problems, improve lives, and create a better future.
I'm with you on that. So to our listeners, we hope this deep dive has sparked your curiosity about the incredible world of cutting-edge science.
And remember, the future is not something that just happens, it's something we create.
So let's use our knowledge, our passion, and our ingenuity to build a future that's brighter, healthier, and more sustainable for everyone.
Well said.
Thanks for joining us on The Deep Dive.
Until next time.